the message box and I'll read it to speaker, or I can turn on your audio, but make sure you've debugged your audio ahead of time. So we're very, very lucky to have Philippe Lemey speaking today. Philippe got his master's in pharmaceutical sciences and a master's in bioinformatics and his PhD in medical science, all at the University of Leuven. He's already written 67 refereed papers. That was actually maybe a month ago that I did that count, so it's probably more now. He's edited this book, and if you're interested in the phylogenetics of infectious disease, I'm sure you're no doubt already aware of his work. His talk today is entitled Phylogenetic Diffusion Models and Their Applications in Viral Epidemiology. All right, take it away.
Okay, first of all, I would like to thank Eric for the invitation and for managing such a nice system. And I also want to apologize for the technical problems we had back in July, which were entirely on my side. So my presentation has a few animations, and I'm not sure how these are going to work out in the system. So I've put them online as a QuickTime movie, which you can click through as well at the website www.phylogeography.org. So if you follow the phyloseminar link, you'll be able to download those. It's quite a big file, but I'm sure you managed to catch up when we hit an actual movie or an animation. So just like Trevor's seminar a couple of months ago, I will be talking about infectious diseases, many of which are clearly recognized as an imminent threat to public health. Now, novel human infections continue to appear all over the world, and importantly, they also have risen significantly over time. And RNA viruses take up an important role here because they have the evolutionary potential to quickly adapt to novel environments and new hosts. So there is a tremendous challenge in combating viral emergence and their disease burden. And we have been interested in how we can contribute to this by looking at the gene sequences of these pathogens and use those to reconstruct their spread through time and space. So we'll be stepping into the field of phylogeography, which you all know is a discipline concerned with the various relationships between gene genealogies and geography. Now, John Avesi and Wesley Brown are considered to be the founding fathers of this discipline, and they were mainly interested in how geographic distribution of genealogical lineages impacts processes like speciation. A more general goal of such analysis would be try to elucidate the historical processes that are responsible for the current geographic distributions of individuals. And traditionally, there has been a strong interest in how population structure may impact genetic processes, even before we began to extensively sample genetic data from those populations. Now, population geneticists formalize different scenarios of how populations may be connected by gene flows, such as these island models of migration, stepping stone models, isolation by distance, and meta-population models. And the analysis of such models has greatly helped our understanding of how geography and limited gene exchange can impact patterns of genetic variation. And nowadays, I think we can say that there are three different approaches to study gene flow. There is a graphical approach, well known as nested clade analysis, which gained a lot of popularity in molecular ecology. Now, this method has largely been discredited now, but I don't really want to go into that debate here. The population genetics approach has developed over time into the genealogy-based coalescent framework. Now, these take advantage of both the gene tree-based phylogeographic approach and the more traditional mathematical approaches, but now taking full advantage of the data. And there are various implementations of structured coalescent models. Trevor has nicely demonstrated how useful, for example, migrate can be for these type of analysis. Now, it's interesting to know that in several of these population genetic approaches, the tree is often hidden. In fact, traditionally, population geneticists usually concentrated on data with relatively low divergence levels, making the recovery of the true tree nearly impossible. Now, for viruses, we're doing a relatively good job at recovering trees because, again, as Trevor has pointed out, we are generally dealing with diverse and very informative genetic data. In addition, different processes of viral epidemic spread can lead to different phylogeographic patterns. And it somehow seems easier to discriminate between those by explicitly modeling them on a tree. But for a long time now, phylogeographic analysis in virology and in molecular epidemiology has been limited to interpreting the geographic distribution at the tips of the tree. Perhaps it's some associated test of where there is significant clustering according to the location of sampling. But if we really want to gain more insights into the processes that gave rise to the distribution at the tips of those trees, what you want to do is to estimate the parameters that govern this process. And we need to analytically reconstruct where the viruses resided at the internal nodes of those trees. Now, ignoring the parameter estimation interest for a minute, one popular way to reconstruct the location state at the ancestral nodes on a tree based on the location state at the tips of the tree has been parsimony. As a heuristic, parsimony owes a great deal of its popularity to being fast. It operates on a single tree estimate, and by ignoring some sources of uncertainty, it seems to give precise reconstructions. Fortunately, there are also some probabilistic methods available, both likelihood and Bayesian development, for these types of ancestral reconstruction problems. And not surprisingly, they do a better job in accommodating the uncertainty of such inference. Probabilistic methods consider diffusion between discrete locations as a continuous time Markov chain characterized by the stochastic rate matrix. So this rate matrix is essentially the same as a nucleotide substitution matrix, and it carries all necessary information for a complete description of the substitution process among the locations. And although this model has been used before, also as Bayesian implementations, they could be criticized because the data may yield inadequate information to infer all instantaneous rates of movement between the pairs of locations. So we're essentially looking at a single data column, a single observation, to try to estimate all parameters here. However, if you will use this in a Bayesian framework, there's a couple of things that we can do to prevent against high variance estimates of the diffusion process. First, the sampling locations here are certainly not going to be equidistant in reality. So we can take advantage of the possibility to specify priors, and we can derive an informative prior for the diffusion rates based on the geographic distribution of the locations. So we have proposed a gamma prior on the relative rates with means and standard deviation inversely proportional to the distances between any two locations. So in this case, the closer the two locations, the more diffusion we expect between them a priori. So in this way, we can start to incorporate some information that traditionally remained unused. Another thing we can do to take into account the fact that most of the possible location transitions will probably never be observed in reality is to use a Bayesian stochastic search variable selection procedure. So this is a procedure that traditionally applied to model selection problems in a linear regression framework, where we are interested in identifying which subset of potential predictors optimally explains most of the variance in this n-dimensional outcome variable. Now, given the potentially high correlation between those predictors, deterministic search strategies tend not to find this optimal set unless we explore all possible subsets. But this is generally computationally impractical. So recent work in BSSVS tries to perform this exploration in two steps. So in the first step, we will augment the model state space with these indicator variables, these deltas here. And we impose a prior on the regression coefficients that has an expectation of 0 and a variance that is proportional to a p times p matrix with these deltas as entries. So if a delta is 0, then the prior variance on that particular coefficient shrinks to 0, and it enforces the coefficient to be 0 in its posterior. And then in the second step, we can use MCMC to explore the joint state space of these deltas and these coefficients. So in analogy to this approach, we can augment our rate matrix with indicator variables, which allows these rates to shrink to 0 with some probability. And we have opted to specify a truncated Poisson prior on the total number of non-zero rates that prefers to explain the spatial diffusion using a parsimonious set of trees. So basically, we ask from the data, what is the minimal set of rates we need to explain the phylogeographic diffusion process on a tree? Now, this distribution is truncated at the minimum number of rates we need to keep the whole migration graph fully connected, and with rapidly decreasing probabilities towards higher non-zero rates. So in this case, this would be, the minimum would be 3 to keep all these 4 locations connected. Now, this will be a useful approach to formally test epidemiological linkage between locations, because we know the prior expectation for each rate to be 0, and we are estimating the posterior expectation using BSSVS. So we can employ standard Bayes factors to assess which rates need to be invoked to explain the diffusion process. So these models and efficient samplers have been implemented in BEAST, and adding spatial models here in BEAST allows us to use a flexible array of models, including the relaxed clock models and flexible demographic priors. And also importantly, it does not require us to condition on a single tree, so we can average over all plausible trees for our data. And this data is now both the sequence data and location state data. So for this discrete model, you have to specify an additional stochastic rate matrix for locations with its traditional phylogenetic likelihood. So let's have a look at a particular example. When we first started to play with these models, this seemed like one of the most alarming infectious disease problems. Influenza A H5N1, which can cause illness in humans and many other animal species. A bird adapted strain of H5N1 called highly pathogenic avian influenza virus is enzootic in many bird populations, especially in Southeast Asia. And one particular strain has spread globally after first appearing in Asia. So we revisited an H5N1 dataset previously analyzed by Rob Wallace and colleagues. They collected 190 hemagglutinin and neuraminidase sequences, sampled over almost a decade of in different locations in Asia. So the trees shown here are typical time-measured trees inferred by BEAST. And although the evolutionary rates for the two segments are very similar, you can see that the timescale for the NA tree is about twice that of the HA tree. And this can be attributed to higher divergence of this basal clade here, which is all sequences sampled from Hong Kong, which turned out to be a reassortant lineage. So based on the discrete model inference, we can annotate modal states on this tree. So the most frequent state inferred for each node according to the complete posterior distribution. And we can start investigating diffusion patterns across the tree. And you can see here that this magenta color here, which represents Guangdong, seems to be the dominant location throughout the evolutionary history, giving rise to many lineages in other locations. Now, if you look at the posterior probability distribution for the root state, and you can see that Guangdong and Hong Kong in red here share most of the posterior mass in the HA tree. In the neuraminidase tree, however, all posterior probabilities are much closer to their prior probability, although Hong Kong and Guangdong are still marginally higher. Now, this difference is due to the tree height here, which significantly increases the uncertainty on the root state, which, in my opinion, demonstrates an important difference with respect to parsimony inference, which ignores these tree lengths and just wants to come up with one single state exchange to explain this. Now, the fact that all locations get a non-zero probability at the root here is probably a consequence of the data not being informative enough to obtain precise estimates. And this is really where the BSSVS comes in. This can shrink all these probability towards zero. Now, using these, we are able to estimate the spatial diffusion process on real timescales because our phylogenies are also scaled in time units. So we have worked on some ways to visualize the inferred diffusion process using virtual globe software. This is going to be the first animation. And here you can see an origin in Guangdong or Hong Kong, from which it slowly spreads to other locations in China. And then in a few different ways, it rapidly spreads to other locations, including Indonesia, Japan, and in Eurasia.
Can you tell us what the lines and the shading mean? What does obviously the lines mean that there's some sort of transmission going on, but the shading.
So the lines are basically phylogenetic branches. So it's basically a tree projecting onto this virtual globe. So, and the circles here represent the number of lineages in the tree that maintain a particular state. So if we would slice a tree and see how many branches maintain a particular location, then this would represent these circles here. As mentioned earlier, by estimating rate indicators using BSSVS, we are able to construct a base factor test to evaluate each rate, so each connection between all pairwise locations. And in this figure here, you can see all reasonably well-supported rates, which were quite consistent among the two different segments analyzed. Now, while we were looking into H5N1, something more interesting occurred in terms of influenza transmission in the Americas. And with a little exaggeration, we were suddenly dealing what seemed to be a more serious problem. And we followed this swine flu epidemic with great interest because researchers really have been sequencing this epidemic massively and almost instantaneously. So during the first days that people started sequencing these things, you could go every day to GenBank and there were new sequences being submitted. So for us, this was a great opportunity to test our methods. And indeed, if we apply those methods, we seem to confidently infer an origin in Mexico, represented by the green branches here. And then there is a sudden shift from green to the orange reddish part of the tree here, which is locations in the US, and from which it takes off as a pandemic. So again, we can look at this using a spatial projection. And this is another one of those examples that you can interactively play with at Phylogeography.org. So you infer an origin in Mexico from which it expands, and with multiple independent events it's introduced in the US, from which it diffuses to Europe, Southeast Asia, Australia, and New Zealand. And by that time also South America got its viruses. So these are basically summaries from analysis that are based simply on genetic data when and where they were sampled. So enough about influenza for the moment. I would like to switch to some more recent work on rabies, which was done in collaboration with people from Pasteur Institute in Paris. Rabies is a viral zoonotic neuroinvasive disease that causes acute encephalitis in mammals. And there are still about 50,000 human deaths annually occurring from rabies worldwide. And most of these occur in the developing world, where the primary reservoir is the domestic dog. And I think it's quite intriguing that an acute and mainly fatal disease can achieve prolonged endemicity, and we think that the spatial dynamics may play an important role in maintaining this virus. So to investigate this in a bit more detail, we focused on a dense rabies sampling in North Africa, including Morocco, Algeria, Tunisia, and some of the Spanish territories in North Africa. Now, our collaborators obtained almost 300 sequences, each encompassing about a quarter of the rabies genome. And using BEAST, we estimated a timed evolutionary history here, which places the most recent common ancestor of all these viruses somewhere late in the 19th or early in the 20th century, which seems to give some support to suggesting that rabies became enzootic in this entire region during the 20th century. Now, interestingly, this analysis also revealed distinct phylogenetic lineages here in Algeria and Morocco and Tunisia, with only a few exceptions in these clades. So this indicates that these viruses generally group according to their country of origin. And we did not really expect this because this virus to respect geopolitical boundaries, because it's supposedly dispersed through rather local movement of animals as observed in wildlife rabies. Now, if you look within a country, there is still some structure according to sampling locations. We're looking at 29 different sampling locations in this tree. But the diffusion is far more dynamic at this scale. You can see much more intermingling of differently colored lineages here. So to test what may have driven rabies diffusion in the specific countries, we tried to encode several potential predictors as rates of diffusion. So any predictor of diffusion that can be expressed as a pairwise measure between all pairs of location can be used as fixed rates in the CTMC matrix. And equal rates can be used as a reference here. So we also considered great circle distances, road distances, which represents the shortest path, the shortest road path for traveling between any two locations. We also looked at spatial accessibility estimates, and these data reflect the travel time to the nearest major city using road and track-based travel. Now, to translate such a continuous accessibility landscape into an origin distance matrix, so basically trying to get this landscape into pairwise distances, we can use what people call circuit theory, or the so-called isolation by resistance model, which has been proposed by McRae. We also tested a potential role for population sizes. But in the absence of real dog population sizes, we had to resort to human population sizes as a proxy. And finally, distances and origin destination population sizes can be combined also into a simple gravity model. So we fitted all these models to the separate Algerian and Moroccan sequence data and evaluated their fit using marginal likelihood estimates. And these are summarized in this table here. And you can see that the rabies spatial dynamics are best described by road distances, which is consistent for both Algeria and Morocco here. Now, distances, just simple great circle distances, are only marginally worse, but there's also a high correlation between road distances and geographic distances. So the virus seems to respect administrative borders while having a relatively fluent dynamics within a country, and there is a good fit between these spatial dynamics and road distances. In addition, if you look at distances covered according to the expected state transitions and their associated distance on branches that represent short evolutionary times, we also noted occasional long distant movements with distances up to 700 kilometers covered in less than 2 or even 1 year here. Now, such estimates seem to contrast with known estimates of spatial rabies movements in African domestic dog populations, which to us seems to suggest that there is some role for human-mediated dispersal of the virus. So, to investigate more formally how the rabies distribution we observe in Algeria and Morocco contrasts with the patterns of spread we would expect from natural transmission dynamics in African dogs, we simulate a sort of a phylogenetic process along the posterior distribution of the trees that we inferred, based on epidemiological parameters obtained from detailed analysis of rabies transmission. So, thanks to a study by Katie Hampson and colleagues, we have a very good quantification of case-to-case transmission in Africa, both with respect to the incubation time and the infection time, as well as the distance between source and recipient dogs. So we simulated chains of infections along the branches based on these parameters and move each new infection with time t in a random direction with a distance drawn from this distance kernel for direct dog contact here. And if we do this recursively in the tree from the root, this leads to location realizations at the tips, which we can then summarize for the complete posterior distribution. So if we perform such simulations for Algeria, starting from the center of all sampled locations, which are shown here as little purple points, we achieve a spread that only includes a limited number of locations. So this is the full white circle here. The increasingly transparent surfaces represent distributions that were obtained by enforcing successive infections to move consistently in the same direction. And even if we do this up till a year, which seems highly unlikely for dog movement, we cannot cover our empirical distribution. And the same is true for Morocco. So these simulations clearly show that rabies could not have spread to the same extent as shown by the current sampling in Algeria and Morocco if the virus was simply being transmitted by natural dog dispersal. So in conclusion, all our analysis for this rabies dataset seemed to point at a contribution of human intervention and displacement of rabies infected dogs by humans, which we think is important to consider for intervention strategies. So the rabies analysis also demonstrates that we can take advantage of the possibility of specifying priors or fixing parameters in the matrix to test different predictors of viral diffusion. However, we basically need to fit every scenario independently, and we cannot really cover hypotheses that would include different contributions of different possible predictors. And Mark Suchard quickly realized that this is essentially a problem that can be addressed using a generalized linear model approach. So recently, we have worked on such a GLM extension of the discretized diffusion model, in which the log of every rate of movement is a log linear function of several potential predictors. So here the betas represent the weights of every contribution, and the deltas, or weight indicators, that, similar to the BSSVS approach that we discussed, enable us to identify the most relevant factors in pathogen dispersal. And also importantly, this parameterization is far less parameter rich than a simple discrete model. So to demonstrate the usefulness of this approach, we can go back to the rabies results, which we already discussed in terms of marginal likelihood estimates. Now in this table, I also added the GLM results for Morocco. Note that this does not include equal rates, because the GLM reduces to equal rates when no predictors are selected in the model. We can now also include separate origin and destination population sizes as separate predictors. And the model also can include a gravity model as a hypothesis, because it can include both population sizes and distances. So we can see here that the road distances are included in the model in about 90% of the time, whereas other factors obtain low or virtually no support. Also, the coefficient estimates conditioned on the predictor being included in the model only exclude 0 for the road distances. Well, their credible intervals only exclude 0 for the road distances. And only for this factor, there's a high base factor support. So in general, these results are very much consistent with our marginal likelihood estimates. But we now have an approach to efficiently explore all possible subsets of predictors in a single run. So we also started applying these tools to human influenza H3N2, the flu variant that dominated most seasonal epidemics since 1968. In particular, we looked at a large data set of about 1,400 sequences sampled globally between 2002 and 2006. And this was published by Colin Russell and colleagues. And this is also the data set that Trevor looked at and explained us in his phyloseminar. Now, for global influenza dispersal, air traffic is an obvious predictor, but this has never really been formally demonstrated. So we wanted to test this using our GLM approach. And thanks to Dirk Brockman from Northwestern University, we were able to collect air travel data between all the relevant sampling locations for this sequence data. And we tested this together with distance, population sizes, origin and destination population sizes, and also origin and destination latitude. And we noted that only distance and air traffic represented by passenger flow were consistently included in the model. But this one, the air traffic, made by far the largest contribution to the diffusion dynamics. Now, by exploring antigenic and genetic progression over time, Colin Russell also suggested that newly emerging strains of this subtype appeared in East and Southeast Asian countries, whereas other locations, such as South America, were mainly evolutionary trailing. And we revisited this question by estimating the time the virus spends at particular locations along the backbone of this tree, the backbone or the trunk of this tree. Now, Trevor already explained us that from a source sink dynamic perspective, this notion of a trunk can represent the source population from which new lineages are expected to be spawned over and over again. So if we summarize these what we call dwell times along this backbone for the complete posterior distribution, we obtain these results for the locations involved. So here the locations are ranked according to longitude, and they are colored in by latitude. And largely in agreement with the findings of Russell, we established Southeast Asia and China in particular. All these we subdivided China, which had a lot of samples, into 4 different discrete states, as being the source population along in this backbone. So we haven't really looked into detail into time slices yet. So particular exceptions to this, as Trevor pointed out, might still exist.
So that right there was showing the location of the backbone, like what percent, what amount of time.
Yeah, so this is basically what amount of time it spends in the backbone. So this should be time in years. So most of its time seems to reside in East China. So I hope to have shown that this discrete model is quite flexible to do phylogeographic hypothesis testing. However, sampling schemes in continuous space may not always fit this discretized diffusion model very well. And the discrete model also assumes that ancestral viruses at any point along the phylogeny here reside in locations from which we actually drawn samples at the tips. So this can be quite unrealistic in many situations. So for this, spatial diffusion in continuous space presents an attractive alternative. For continuous traits, in this case, latitude and longitude, Brownian diffusion is the sort of the random walk analog to the Markov chain transition model. And such bivariate Brownian random walks have already been suggested in a likelihood framework. And we've recently implemented these in a Bayesian framework as well. So these can now be used to infer continuous diffusion processes in space and time. And for the continuous model, we can take conjugate priors over the root location and the precision matrix, and again formulate a traditional phylogenetic likelihood. So with these tools, we can start to produce estimates of phylogeographies with continuous location realizations and times for each node, such as this raccoon rabies example, for which samples were taken during 3 decades of epizootic in northeastern United States. So again, this is simply a phylogenetic tree produced by this model that is projected onto a virtual globe. So every location is shown according to its, every node in the tree is shown according to its inferred location. And the height represents the time. So again, we can visualize those in more detail. And this time we can represent uncertainty by slicing the posterior distributions of trees and contouring the imputed locations at a particular time point along the branches that are sliced. So if we do this, you can see an origin here and expanding over time. So again, these blobs represent sort of the 95 credible intervals for the inferred location or the imputed location along branches. Now, we all know that Brownian diffusion is a very restrictive process, mainly due to an implicit assumption that the process cannot vary over time. Now, to overcome this limiting assumption of constant diffusion rate through time, we can borrow ideas from uncorrelated relaxed clock models. So essentially, we can allow heterogeneity in the process by drawing for each branch in the tree a scaling factor from some underlying distribution and rescale the variance accordingly. So this is basically using the same models that Alexey Drummond used in his relaxed clock paper. So if we relax the constant variance walk, we can also get more realistic estimates of dispersal rates through time. And these are shown in blue here. And we can see that these dispersal rates are relatively high and increasing during the initial phase of the epidemic. And it attains values up to about 26 kilometers per year. And then it declines and stays at relatively low rates in this initial phase, in the further wave. So really the expansion, the spatial expansion, has occurred in the first decade. And after that, most of the diffusion has been mostly local. And this is superimposed onto one of these skyline type of plots, which should be a reconstruction of the demography through time. And if we want to overinterpret this a bit, you could see that there's some sort of relationship between the expansion in terms of space and population size over time.
I don't quite follow the point, that last point you wanted to make it.
Well,
I see that this one goes down and then.
Well, if you can see if it initially it covers large distances, and with a lag, it seems to increase in population size as well. Now, when the dispersal rate goes down, the population size stabilizes, and now I'm really overinterpreting this a bit. You can see a bit of an increase in dispersal rate at this time point, which coincides with another increase in population size. And again, when it goes down, the population size stabilizes. But I really acknowledge that I have to do a bit of overinterpreting here. So in our opinion, relaxing the diffusion process is really a much needed extension, because it may prove difficult to find any real-life example of spatial diffusion that really adheres to a homogeneous Brownian diffusion process. And in this West Nile virus example, on which we worked with Oliver Pybus from Oxford, we observed so much rate variation in the process that it really becomes a challenge for efficient MCMC integration. So if you look at this reconstruction, you can see both rapid long-distance dispersal as well as more local, slow diffusion processes. Now, an important direction we would like to take with this in the future is to find ways to accommodate spatial heterogeneity. So, in addition to relaxing the time homogeny of the process, we also like to build in some spatial heterogeneity, and I think then it would become very useful for hypothesis testing as well. And finally, I would like to conclude with a little summary on how our phylogenetic diffusion models relate to other methods. So the comparative approach may be useful to assess geographical spread from molecular data, but without modeling the spread explicitly. And we know that there are some ABC developments that try to do this in a more rigorous fashion than NCPA. If one wants to get information about population sizes and migration, but one considers population to be really a coarse feature, and one is willing to make abstraction from the spatial dimension, then population genetics approaches will probably be very, very useful. Now, if one wants to model rates of spread and explicitly take into account geographical features, then I hope that the methods presented here may be of some use. We also acknowledge that there are still some open issues for the application of such methods. I think sampling design and potential biases is no doubt the most important one. And just as a last note, I would like to add that these diffusion models are really general models of trait evolution. So any trait that one is interested in can be accommodated by these models. For example, Andrew Rambo from Edinburgh University is currently applying these models in collaboration with Derek Smith and Colin Russell to try to integrate influenza genetic and antigenic data. So those who have seen Trevor's talk will probably remember those antigenic maps that he showed. Well, with these models, we can start to examine in more detail how these maps, how this antigenic evolution evolves through time. So our visualization methods are still limited to Google Earth, but sort of to make abstraction from the geography, we plotted this on the moon. And you can see that there's clearly different phases of exploring antigenic phase. So it goes from left to the right here. But at each, there's sort of 4 different periods, and at each period it tries to explore antigenic space around the central point.
Can you just say a little bit about what that projection is?
So basically these serological tests give you a lot of antigenic values for each virus that is sequenced. And then you can do some sort of multi-dimensional rescaling to get 2 principal components of antigenic space. And we're basically modeling those two principal components as a bivariate random walk onto the sequence tree. So, you know, these maps, these antigenic maps that have been published, kind of show some nice endpoints of these processes. But now we can start to connect those points, and we can start to test hypotheses whether, you know, this type of antigenic evolution occurs more rapidly along the backbone, or other types of scenarios that one can come up with. And of course, I would like to thank many people who were involved in this work, in particular Mark Sucher and Andrew Rambo. Mark has been instrumental for most of the developments presented here. And I would also like to take this opportunity to advertise a postdoc position on an ERC grant to work on these type of methods and applications. And would welcome any questions.